Right. Good afternoon, everyone. Thank you so much for the introduction. At the end of the session, two of you please hire me. um, so I'm a third-year PhD student at the University of Rochester, and today I'll be talking about recapitulating phenotypes of acute myeloid leukemia (AML) in a 3D model of the bone marrow microenvironment, the BMME. Acute myeloid leukemia is a malignant blood cancer that takes place in the bone marrow. And right now, there is an alarming rate of incidence and death rates for AML globally. And patients who have AML, they have a very poor 5-year survival rate of less than 30%. And for patients who are above 65, which accounts for the majority of the diagnosis, the 5-year survival rate is less than 10%. And it's worth to note, within this patient population, 70% of them die after 1 year of therapy. 1 year of diagnosis. These patients have very, very poor outcomes due to the high prevalence in the aging population. These populations, they do not respond very well to standard chemotherapies that are out there. And these chemotherapies can produce high relapse rates. So it is very clear that we need better therapy for AML. AML takes place, again, in the bone marrow, where that is the site of normal hematopoiesis. Normally, there are these hematopoietic stem cells and progenitor cells, and today I'll be referring to them as blood stem cells. These blood stem cells, they differentiate into myeloid and lymphoid lineages, and at the end, they make up our erythrocytes, osteoclasts, T cells, and B cells. During acute myeloid leukemia, what happened is that there's a population of these blood stem cells in the myeloid lineage. They accumulate mutation, and then they become dysfunctionalized, and they overexpand at the cost of healthy cells in the bone marrow. And patients who have AML, they experience bone marrow failure, essentially. Right now, most AML studies that look at mechanism and also drug discovery are focusing on the intrinsic mutations that generate these AML cells from the HSPCs. But less is known about the role of the tumor microenvironment that Carly have talked about before. Less is known about the BMME in progressing AML. So AML and blood stem cells obviously do not live alone. They exist in this very intricate bone marrow microenvironment. And today I'll just be highlighting some of the key components of the BMME. So one of the key components we have are the osteoblasts and osteoclastic cells, these bone-forming and bone-breaking cells shown over here. And then you also have endothelial cells that line up the arteries, capillaries, sinusoids, and you also have stromal cells that provide support for the HSPCs, the blood stem cells and the endothelial cells as well. And there are other various types of cells as well. And we also have that stromal matrix that house all of these together. This BMME, what they can do to the AML is they can protect these cells from chemotherapy, so providing a protective island to them. And not only that, these AML cells can signal through the various microenvironmental cells and change the function of these healthy cells. And in return, after the change of the function, you can generate a leukemia-favorable environment so that can sustain the AML cells and also promote the disease. And overall, when the environment is dysregulated, what happens is that there is a loss of normal hematopoiesis. So you're unable to generate healthy blood cells anymore. And the loss of normal hematopoiesis associated with this dysregulation is what account for the mortality and morbidity of AML. So right now in the field, the critical need is for us to understand how the overall BMME can support AML cells and can support disease progression. However, obviously, the current gap is that there's a lack of reliable in vitro model that can fully recapitulate the overall BMME interaction with the AML. So our lab is a cancer microenvironment lab, and we've studied a lot from the various papers that have been published from Donald Ingber's lab, Krishnendu Roy lab, and whatnot. And we decided to use the MPS approach as well. So to start, we created a murine bone marrow microenvironment on a chip using the amyloid chip S1. And what we're trying to do is recreate the architecture of the BMME. So first of all, we generated an osteoblastic niche. So we seeded stromal cells that we get from mouse onto the top channel, on the top surface of the top channel, and then we allow for osteogenic differentiation for about 14 days. And at the end of that, we can fix the osteoblastic layer and stain for markers of mature osteoblast activity. So this data is showing alkaline phosphatase staining and also von Casa staining. Von Casa stains for all these darker nodules. If you can see these dark spots, these are the calcium deposition that the osteoblasts give. And then all these pink staining are markers of alkaline phosphatase positivity. So after that, then we want to generate these sort of the vascular mimic. So we seeded endothelial cells in the bottom channel of the amyloid chip. And these form monolayer shown by tight junction formation Z01. The third component is the fibrin hydrogel to provide a 3D matrix. And within this 3D hydrogel, we encapsulated more stromal cells that are known to support the blood stem cells. And then we also encapsulated blood stem cells that we sorted from mouse. We cultured this device for 14 days with continuous flow in the basal channel of this chip. And then at the end of that culturing period, we can harvest for multiple output. So without belaboring the data, you can refer to this publication that we published recently. What we have shown is that our blood stem cells are maintained at a similar level to native bone marrow values. And when we took the cells from the chip and then we injected them into mice that have been irradiated, so they have bone marrow failure, these cells can engraft into the bone marrow and repopulate the bone marrow cells and also repopulate blood populations as well. So here is a snippet of the data. We can see that our chip cells were able to repopulate the monocytes, B lymphocytes, and T lymphocyte population. Now moving on to now generating our AML on chip. So in our lab, we use this clinically relevant murine model of AML. It's called the blast crisis chronic myelogenous leukemia model. In this model, we take blood stem cells and then we transfect them with two sets of genes, BCR-ABL attached to GFP and NUP98 HOXA9 attached to YFP. And these two sets of genes, they are able to create a leukemia phenotype in these mouse. What happened to this mouse when we look at the bone marrow, you can see an increased frequency of blood stem cells. So here is just showing LSK, its marker for the blood stem cells, lineage negative, SCA1 positive, and c-KIT positive. They are expanded in the mouse, and then more more naive populations, long-term and short-term, repopulating blood stem cells. There's also an expansion of these two populations. And in this mouse, what happened is that you get expansion of these blood stem cells, but they're not differentiating into mature blood cells at the end. So one other important output that we looked at is looking at osteoblastic function. So again, osteoblasts are the bone-forming cells. And what happened in this mouse is that you see a reduction of these markers of osteoblastic activity. So here is just showing in situ hybridization of osteocalcin, all these blue spots over here. And you can see after 10 days of initiating the disease, you get marked reduction of osteocalcin staining. And that trend is seen similar with collagen 1 activity as well. And Ben, my PI, when he was working with Laura Calvi's lab at the University of Rochester, he has shown that this dysregulation of the osteoblastic cell function is due to this chemokine CCL3. And CCL3 is overexpressed in murine, and it can inhibit osteoblastic function. More recently, when we knock out CCL3 in the leukemic model, we can see that these mouse, they survive past 25 days for at least until 45 days. So normally they would just die at 25 days. So we're very excited to study this chemokine in our system. So moving on to generate our AML on chip. So we took 70,000 of these leukemic cells, they're GFP positive, and then we combined them with 70,000 whole bone marrow cells, and we added them into the bone marrow compartment of our chip. And then we can culture, for this experiment, we cultured for 7 days and perform multiple analysis. And after 7 days of culture, you can see all these GFP plus AML cells on the top channel. They still survive. These bottom channel over here is stained with DAPI, and that's the endothelial cells. We looked at flow cytometry first and look at the population of these AML cells, and we can see that they're at about 30% compared to our wild-type chip. There's no AML cells in there. And this is important because in advanced stages of AML in patients and in mouse, you see about 30% of AML cells in the bone marrow. Another output that we can look at is gene expression. So we performed qPCR, and here we looked at osteocalcin and collagen 1A1 of osteoblastic activity. And what we can see is that in our AML chip, comparing to the wild-type chip, there is a marked reduction of osteocalcin. And we don't see a reduction of collagen 1A1, but that might mean that we need more longer time point to look at the change of this marker. So another thing that we look at is mineralization of these osteoblastic cells. What I did with this experiment is I flushed out the gel and then fixed the remaining osteoblastic layer that are on the chip. And I stain again for alkane phosphatase and von Casa staining. And it's a little bit harder to tell with the pink staining, but if we take a closer look, we can look at the calcium deposition, all these dark spots, and you can see in our wild-type chip, there's lots of calcium nodules, but looking at our AML chip, there's reduction of these calcium spots. So that means that the osteoblastic cells are losing their capability to deposit calcium. I performed image analysis just using image J, and what I found is that their average nodule area between these two groups do not change, but the total nodule area, so combining all these dark spots, I see a trend of reduction in our AML chip. And more strikingly, when I count the number of these dark spots, you see a significant difference, significant reduction of these spots in our AML chip. So taking the qPCR data and the histological data together, we see that there is something that's happening to our osteoblastic cells. These AML cells, they induce a loss of osteoblastic activity. So that's similar to what we've seen in our in vivo experiments. Another output that we looked at is CCL3 levels in the effluent of AML chip. So as Carly mentioned before, we can take the effluent easily from the reservoir of these chip. And from this experiment, I took effluent from day 0 to day 4, so they're combined, and then day 5 to day 7. And as you can see in this graph here, there's a significant elevation of CCL3 in our AML chip. And I'll tell you why this is important later. And now I want to look at flow cytometry analysis for these chips. So in this experiment, we have, again, 70,000 leukemic cells, and we added a whole lot more bone marrow cells into the marrow compartment. And we can perform flow analysis at day 7, day 14. Today I'll just be showing data on day 14. So first of all, similar to what I've shown before, we look at the presence of GFP plus cells. So at day 14, these cells are at 30% of total live cell population. That's good, that's what we want. And then looking at more immature population with flow cytometry, first of all, we see a reduction of lineage negative cells. So these cells are not your osteoblasts or not, they are your immature cells. And in this data over here, we see that there is an expansion, there's an increased frequency of our LSK, the blood stem cells in our chip. So this is similar to what we see in vivo before. Within the blood stem cell population as well, we can look at more refined, more downstream markers. Here I'm showing MPP4, which is a marker for lymphoid progenitors, and an MPP3, which is a marker for myeloid progenitor. And these two populations have increased frequency in our chip. So the blood stem cell data and then these progenitor data are telling us that in the leukemic condition, there is expansion of these populations, so similar to what we see in vivo, but they might not be able to differentiate into mature blood cell population. So we still have to perform that experiment and analyze the data for flow cytometry, but we're very, very excited to see these phenotypical changes that we've seen in vivo. So to conclude this talk so far, I've shown that in our AML on chip model, it has been able to recapitulate some of the key phenotypes that we've seen in vivo and that is validated in human cases. And then in particular, we see this elevation of CCL3 in our AML chip. And Ben has shown before that when you look at the blood of AML patients, you see an elevation of CCL3, and then more recent groups in 2020 have shown that this CCL3 is also implicated in human cases. So the next step of my project is then to generate the human version of AML on chip, now that we validated what we've seen in mouse into our mouse chip. And with this human chip, hopefully we'll be able to investigate the specific interactions between the AML cells and the microenvironmental cells, especially the osteoblasts. So with that, I want to thank everybody who has been supportive for my research, my PI, Benjamin Frisch, my co-advisor, Dr. Dana Benoit. At University of Rochester as well, we created this Rochester MPS network for trainees to help us support each other and whatnot, and it has been a valuable asset for me. I want to thank the committee, and also want to thank the Travel Grant Committee for giving this opportunity to travel here. So I'll take any questions.
So, really, thanks, Azmeer, for an excellent presentation. We have time for one question.
Just a quick question. I couldn't remember if you had, when you were characterizing your bone marrow microenvironment, did you look at neutrophil production? I might have missed it.
No, we have not done that, but that's very interesting because one of the CCL3 might recruit neutrophils into the cancer microenvironment. We haven't looked at that before, but I think in the future we will definitely do that.
So I think because of time, I think we need to move on. So thanks, Azmeer. And I'm sure if there are any job opportunities, people will come up to you afterwards.